Today we are going to discuss about the structure of bacteria. Basically, structure of bacteria, there was a scientist named as Anton van Leeuwenhoek. He was the first scientist to saw and describe the life cell and the life cell in water. So basically he was the first scientist who saw as well as he saw and describe the life cell. Now basically he has observed many things in the basically he observed the cell wall. So he has divided it into a parts, means on the basis of structure he has described it. He has described the bacteria on the basis of structure. Okay, so first is he has described was a covering portion, and covering portion means like envelope. Okay, so basically he has described three things in envelope. First is glycocalyx, second is cell wall, and third he has described a cell membrane in covering portion. Okay, first is glycocalyx. Calyx basically means a covering, and it is a outer covering. We'll be discussing all these in a detail. First of all, just an intro that he has described a glycocalyx, that is basically means a covering, and glyco is what comes from a carbohydrate. So basically glycocalyx, that is a covering portion and having a cell wall. Now glycocalyx is basically exist in two forms. We'll be discussing these two forms in detail as well. First is capsule, second is slime. Then the second is cell wall. That is basically defined on the basis of staining purpose. First is gram positive and second is gram negative. Like that is having a stain. If in the result we get a stain, that is gram positive. If in the result we do not get a stain, that is gram negative. So basically the cell wall is basically defined on the basis of staining purpose. Now the third one is cell membrane. Basically cell membrane, that is a mesosome, and it exists in chromatopores. So basically chromatopores are basically only found in a photosynthetic bacteria, that are only this cell membrane is only found in the photosynthetic bacteria, and that mesosome is found in all. So basically these are the structure based on that he is described, that is on the basis of structure. Second, he has described that is bacterial cytoplasm. Third, he has described bacterial genome, and fourth, he has described a bacterial appendages. He has described four. First of all, he has described these in very detail, the covering, that is glycolysis, cell wall and cell membrane. Now glycolysis is basically exists in two forms, that is capsule and slime, and cell wall is basically on the basis of staining purpose, that is gram positive and gram negative, and cell membrane in mesosome and chromatopores.